Hello, I'm Dr. Azaz from medicovisual.com back with another visual lecture on the topic of development of Telencephalon. Now, Telencephalon is actually the derivative of the cerebral cortex and basal ganglia and some other structures that we will discuss today. Okay, so from the previous lectures, if you haven't watched those lectures, please watch that first because this lecture is related with the other lectures that I have discussed previously. So we had discussed that three primary brain swellings develop. So three primary brain swellings, prosencephalon, then there is mesencephalon, which is basically the midbrain swelling, and then there is the third one is rhombencephalon, which is the hindbrain swelling, and then of course there is spinal cord. Now this one is prosencephalon. So prosencephalon here is shown this as the third ventricle, right? So prosencephalon, its anterior part, its ventral part and ventrosuperior part, especially this part is actually traditionally or conventionally it is considered as a part of telencephalon. The remaining part is considered as a part of the diencephalon, but the ventral part is considered as the part of the telencephalon, right? So now what will happen that from the ventral part, and by the way, these diencephalon and telencephalon both are the parts of prosencephalon. Please don't forget about that. So now from the ventral part, what actually happens that evaginations arise, and these evaginations, they will form the lateral ventricle like this. So you see, let me show you again. So evaginations, bilateral evaginations arise like this, and these are the primordial lateral ventricles. Now these are connected, these lateral ventricles, these are connected with this connection to third ventricle. This connection is called interventricular foramen of Monroe. Now this part, as I've told you, this ventral and ventrosuperior part of third ventricle is said to be derived from this part. It is said to be derived from the telencephalon, not diencephalon. The remaining part is said to be derived from diencephalon, but this part is said to be derived from telencephalon. This part of telencephalon, this middle part, this median part of telencephalon is called telencephalon impar. This is called telencephalon impar, but the other one is forming the lateral ventricle, which hasn't been given a very special name, but you just can call it telencephalon proper. I just devised the name. So anyhow, this is the situation right now. Now of course, with this diencephalon part, it is not just third ventricle. Of course, around that neural tissue develops. So around that neural tissue develops, and in case of diencephalon, we had discussed previously that it will form thalami, the hypothalamus, thalamus, subthalamus, and we have discussed those details. Similar to that, here also in case of lateral ventricle, here also some neural structure, neural tissue develop, and these neural tissues, they will form certain structures that we will discuss today. But before that, what is this red thing? Actually, this is the choroid plexus of lateral ventricle. We have discussed already that how choroid plexus develop, but here let me briefly tell you that if I take a section of this this lateral ventricle here, okay, so this is the lateral ventricle, its section here. So now of course around that will be the meninges, right? Pia mater, arachnoid and dura mater. Now what actually happened that this pia mater is quite vascular at this point near the junction of floor of lateral ventricle to the roof of third ventricle. Actually, the lateral ventricle is relatively near to third ventricle, but here this 3D model is not anatomically correct. It is more of a schematic model rather than being anatomically correct. So of course here, by the way, here there would be third ventricle. So near the at the junction of roof of third ventricle, this is roof of third ventricle, to the floor of lateral ventricle, near its junction at this point. Here what will happen that this vascular pia mater, it will invaginate inward. It will invaginate inward like this, and along with that, of course, blood vessels will also invaginate inward here, right? So blood vessels will also invaginate inward, and of course they will branch, and of course pia mater will invest it like this. In this way choroid plexus is formed. I hope you understood that how choroid plexus develop. And of course choroid plexus here is it's a long network. Here you see it's going on like this, and of course it will follow. We will discuss that later, and by the way, again, we will not go into detail of this. That is the topic of neuroanatomy, but here we will focus more on the neuroembryology, right? So I have told you that here is this lateral ventricle, primordial lateral ventricle. It is not complete lateral ventricle. So here you see that at the cross section, or even if you do not take the cross section, you can see that it looks like more of a triangle, right? It looks more of a triangle. Now again, the neural tissue will develop in its vicinity. Let's talk about that neural tissue. So neural tissue will develop here, and here you see along with that here some neural tissue here. They will aggregate, they will accumulate, they will aggregate, and they will form elevations at the floor of this developing ventricle. So here you see developing lateral ventricle. So what are these swellings? These are let me name these swellings for you because these are relatively important. So this is medial ganglionic. Here the term ganglionic is a misnomer because there is not a ganglia here. So anyhow, conventionally it is called medial ganglionic eminence. This is lateral ganglionic eminence, and this is the caudal. It is behind. It is caudal ganglionic eminence. Now these ganglionic eminences are very important because some of the important structures develop from them. Now along with that, just slightly cranial to this choroid plexus, another structure develop. So this structure here, this is actually the primordium of hippocampus. So here this is primordial hippocampus. Here these are the ganglionic eminences. Now here we will also discuss later in details that from the ganglionic eminences what will happen, that subcortical tissues will develop, subcortical structures will develop. Subcortical structures, for example, lentiform nucleus, that we will discuss later, lentiform nucleus, and especially from the caudal eminence, from the caudal ganglionic eminence, amygdaloid nucleus will develop. Now these are the structure at the floor of this lateral ventricle, primordial lateral ventricle. It is not a complete mature lateral ventricle, it's a primordial lateral ventricle, which is of course the part of telencephalon. So we can say that the cavity of telencephalon is lateral ventricle, and this structure at the medial wall of this developing structure, this structure is hippocampus. Not actually hippocampus. It is, of course, primordial hippocampus. It is not a mature hippocampus. It is primordial hippocampus. Now, beside these two structures, all of the other neural tissue in this area, it will form the remaining cerebral cortex, right, along with the olfactory cortex as well, and even the olfactory bulb. So other part of the cerebral cortex will develop from the remaining structures. And by the way, here I have previously forgotten to tell you that, so you see that this was the Telencephalon impar. Its cranial limit and cranial and ventral limit, ventrocranial limit, this wall or this limit, it is actually called lamina terminalis. It is called lamina terminalis. It is termed as lamina terminalis. So anyhow, now here let me tell you that for most of you people, for most of the undergrad students, and even for those who are appearing for exams like USMLE, this much information is enough that telencephalon, it forms the cerebral cortex and some subcortical tissues, some subcortical structures like basal ganglia and amyloid nucleus, and along with that, of course, hippocampus, which is anyhow the part of cerebral cortex. So cerebral cortex and subcortical structures, they develop from telencephalon. That is it. This is the most important thing, the take home message of this lecture. So it is enough for most of you people. If you are here for the exam point of view, you can skip the remaining part of lecture, but if you want to learn the concept, then you can stick around. But let me give you a heads up in advance that this topic is a bit complicated and relatively low yield. It's not very important from exam point of view, except that you must know that what structures develop from telencephalon, which I have already told you. But its details, these mechanisms, these are not even precisely known, and these are not often asked in exams, so these are not high yield. So but anyhow, I will explain these structures for you, for the concept loving people, so they can stick with me and they can continue to watch this lecture. So enough of this yapping. Now let's get back to the lecture. So now what I will do is that we will discuss the development of these structures one by one. So now I will hide the primordium of cerebral cortex, right? This is the primordium of cerebral cortex that I've just hid. I will hide the primordium of hippocampus, and here I will just focus right now on these ganglionic eminences. Now what will happen that these ganglionic eminences, they will be remodeled in such a way that they will slightly change the position like this, and of course they will change their shape. You see, this is the situation right now. So now what will happen that the lateral ventricle, it will also remodel this primordial lateral ventricle. It will also remodel in such a way that, let me first explain that it will slightly grow ventrally. By the way, this is ventral side and this is dorsal side. So it will slightly grow ventrally, and along with that it will also grow dorsally, and then it will bend and fold like this. So you see, it will grow dorsally and it will bend and grow like this, and then it will form a tail behind. So let me explain it here on a mini diagram. That first there was a relatively straight one. Then what will happen that it will grow forward ventrally and it grows dorsally, and this dorsal one, it bends like this, right? And then here a tail will form. So this will be the situation with the lateral ventricle. And as these things are happening with the lateral ventricle, other structures undergo similar type of remodeling. I mean the lateral, as this one is the medial, by the way, greenish one. The medial ganglionic eminence, lateral ganglionic eminence, they will undergo similar remodeling, and this caudal, it will stick to back side of it will continue to stick to back side of lateral ganglionic eminence. And now let's see how this remodeling occurs. It's very easy to understand. Let's watch the animation. So you see how this remodeling is occurring. Let's watch it again. It's very easy to understand. I don't think it's very difficult. You see, and of course here the tail will form. You see, like this. And if we focus on these structures, they will further remodel. And what about this caudal eminence? Caudal eminence will move at the tip of this lateral. What is this? Lateral ganglionic eminence. And then what will happen, that this will sort of, the lateral ventricle, it will become much smaller. It was relatively huge, but it becomes relatively smaller as the cerebral cortex, that we will see later. As the cerebral cortex, it grows, there is growth of lots of white matter and gray matter. So this lateral ventricle, it is not much needed, so its volume will shrink quite a lot. And this caudal ganglionic eminence, which is the primordium of amygdaloid nucleus, it will come to lie at the distal tip of this lateral ganglionic eminence. And this lateral ganglionic eminence as well as medial ganglionic eminence, they will also remodel like this, both of them, and they will form a structure like this. It's all visual. I don't think I need to explain a lot. I can use a lots of words, that it is twisted like this and it happens like this and that. First thing is that no one will ask this thing in your exam. Second thing is that it is visual and it is conceptual. So just watch this animation. It's beautiful, isn't it? So now what will happen, that the nerve fibers, they are descending downward from the cerebral cortex. In this 3D model, I have not shown the cerebral cortex right now. We will put that in picture later, but of course there is cerebral cortex here, and from cerebral cortex descending fibers are growing and they will grow downward like this. And as it happens, then what happens here, that you see that they have to pass through this lateral ganglionic eminence, which is no more lateral ganglionic eminence. Its name has now changed. Let me tell you what it is now called. So you see that they go through this and they go downward like this. So you see what is happening here, that this internal capsule, which is actually the white matter tract that is going from the cerebral cortex downward, even some upgoing fibers are there. So you see that here, as they are passing through this, some holes develop, some, you see, striations are actually developed. What I mean by striations, that if you focus here, that there is alternating gray matter, and then we have white matter in the form of internal capsule, then gray matter, then white matter. So you see striated appearance is seen here, gray, white, gray, white, gray, white. So you see striated appearance, and because of this striated appearance, this structure is called corpus. Corpus mean body. Corpus striatum, striated body. So this structure is called corpus striatum, which consists of two parts. Further, it consists of two parts. This C-shaped nucleus, on the tip of which is this amyloid nucleus. So this C-shaped nucleus here, which is connected with the help of these connections, these interconnections, especially head is very strongly connected with this other structure that I will tell you what is that structure. So this C-shaped nucleus is very logically called caudate nucleus. C-shaped nucleus, caudate nucleus. So this is caudate nucleus, and the other one is just like the name of some mythological character, I think. So that is called, the other one is called putamen. That is called putamen. So basically caudate plus putamen together, they are called corpus striatum, right? So these are called corpus striatum. By the way, this classification is a bit confusing. Some people say these two are called corpus striatum, but other also include globus pallidus, that we will discuss later, the medially developed structure. Some people also include that in this classification. That is a bit debatable topic, and there is a reason for this classification as well. There is neostriatum. These are lots of terminology. I will not go into that debate and that controversy. That will be taken up in neuroanatomy. For now, if you don't want to go in debate, just remember that this structure, this round one, the spherical one, somewhat spherical or oval structure, that is putamen. The C-shaped structure is caudate nucleus. So these two structures, these are quite interconnected with each other, and they receive the input of basal ganglia. Again, I will not go into physiology of basal ganglia. Actually, for the each and every structure that I am going to discuss in this lecture, this development that I am taking up in this lecture, for that we can create a lecture series on their functions. So right now, please allow me to focus on embryology, otherwise I will deviate into the physiology, neurophysiology and neuroanatomy. So you see caudate nucleus and putamen. And what about this green structure? If I hide this internal capsule, you see this green structure which developed from medial swelling, medial ganglionic eminence. This structure is called, what it is called? It is called globus pallidus or globus pallidus. Yeah, so this is the situation. Now if you focus on the putamen and globus pallidus together, you see this is putamen. I don't know which color should I use. Okay, let's use this one, or yeah, this color is fine. So this is putamen and this is globus pallidus. If you look at them carefully and as a single entity, if you look at them, they somewhat resemble the shape of a lens. Of course, not perfectly, but somehow you can see that they somewhat resemble the shape of a lens, a biconvex lens. So that is why both of these structures together, the putamen, this yellow one, and globus pallidus. So both of these structures, let's write it here. These are called together as lentiform. They look like lens. Lenti mean lens. Lentiform nucleus. Lentiform nucleus, which consists of not caudate but putamen. Caudate is excluded from it. Putamen plus globus. I hope my spellings are correct. I'm not good at spelling. Even my memory is very weak. I forget the details. I only remember the concept. I forget the details. Even I don't remember birthdays or any type of dates. I have very bad memory. Yeah, that is what it is. So yeah, that was a myeloid nucleus. So from this lecture up till now, what we can infer that lateral ganglionic eminence, it developed the caudate as well as putamen. It formed the caudate as well as putamen. The medial ganglionic eminence, it formed the globus pallidus, and the caudal ganglionic eminence, it formed a myeloid nucleus. But these are not only structures that develop from these swellings. Some of these cells from these swellings, they move into the cerebral cortex, that we will discuss in the histogenesis of cerebral cortex. Some of these cells, they move into the cerebral cortex, and there they form inhibitory interneurons. that might be asked in some MCQs, that they form inhibitory interneurons. So don't think that these are the only structures that develop from these swellings. And by the way, these structures excluding amygdaloid nucleus, included in that the amyloid nucleus is the part of limbic system. So other structures, the caudate, the C-shaped caudate nucleus, the putamen and globus pallidus, and some authors even include subthalamus, which is derived from the diencephalon, and substantia nigra, which develop from mesencephalon. So all these structures, they are considered as a part of basal ganglia or basal nuclei, to be precise. Ganglia again is a misnomer, these are nuclei. So basal nuclei, which have very important function in the motor control. They have some motor plans stored in them. I will not go into details of its physiology, but here embryologically you must know that they develop from the floor of lateral ventricle. They develop from floor of telencephalon proper, and specifically from the lateral and medial ganglionic eminences. That is the most important take home message of this part of the lecture, which was about the subcortical nuclei, specifically the basal ganglia and amygdaloid nucleus is the part of a limbic system, which is related with emotions and memories and some other stuff that let's ignore them. So we have discussed about the basal ganglia. Now let's include hippocampus. It's very easy to understand. So let's include hippocampus here. Now the situation with the hippocampus is again very similar, that as the lateral ventricle, it undergoes the remodeling as it is folded. This hippocampus, primordium of hippocampus, it will also fold like this, as we have discussed already. It will also fold like this, and here, yeah, so it will also fold like this. But interesting thing that happens here with the hippocampus, that this is the proximal part. Let's say this is the proximal part, and this is the distal growth, not outgrowth, distal development or distal further growth of this primordial hippocampus. Now this proximal part, it will mostly degenerate, not completely, but it will remain as a very narrow strip of gray matter. It will mostly destroy, but it will only a part of it will remain as a narrow strip of gray matter. But this part, this distal part, which is near the temporal lobe, here you will learn that temporal lobe will develop here. So this part remains and this part thickens actually, but the other part, it is mostly attenuated like this. Let's watch the animation. So you see this part is attenuated. So this part is attenuated, and this attenuated strip of gray matter, which is mainly considered as a vestigial remnant, this is called, that has got a very fancy name, to be honest. That is called indicium griseum. I'm not perfect at pronunciation, but let me write it for you. But before that, let me show you that. So indicium or indicium griseum, it's very fancy name anyhow. So indicium griseum develops from that, and hippocampus proper as well as dentate gyrus develops here. So you see these structures develop from this distal part. So here we have this purplish one is hippocampus proper, and this one is dentate gyrus, which have teeth, not actual teeth. It looks like that it has teeth. So this is dentate gyrus, right? So dentate gyrus and hippocampus proper, and this one is indicium or indicium griseum. Narrow strip is indicium My spellings are not perfect again, sorry for that. Grecium, I think that's the, so that is it. And this one is dentate gyrus. Dentate gyrus is actually connected with this endesium grecium, and this is hippocampus proper. Hippocampus proper. Along with that, of course, in neuroanatomy, you will study there is parahippocampal gyrus, there is entorhinal cortex, there is subiculum, and some other structures. But in this lecture, I will keep it simple and I will not go into those details because that will become quite confusing and lecture will become very large, very long actually, not large, very long. I mean, English is not my first language, so pardon. So now what happens that of course we have the structures that are derived from diencephalon. Let's put those as well here. So these are the thalami. This bluish one are thalami. This is subthalamus. These are subthalamus. This is hypothalamus, and here I have removed the pituitary stalk, and these are the boobs like things below the hypothalamus. These are called mammillary bodies. Yeah, so these are the structures. Now what happens actually that from the let's explain it at this point, that from the this end of hippocampus proper, the nerve fibers arise. Actually not exactly from the end, there is something called alveus, but again I don't want to confuse you. So some fibers are going like this from the hippocampus proper, and they will join to form what we call as fornix. Fornix is white matter bundle that arises from the hippocampus proper. So this white matter bundle, you see it is arising here. This white matter bundle, it will go towards the mammillary body like this. It will go towards the mammillary body, and you see that these are the fornix, and right and left fornix. They will sort of, you see, they will cross with each other. They will crisscross with each other. They will talk with each other. And in this way, the structures that are talking with each other, these are interconnected structures, right and left hippocampus proper. They are interconnected with each other with the help of these fornices. So these interconnected parts of the fornix, these are called hippocampal commissures. And of course, we have this indusium grecium. And indusium grecium is here connected with septal nuclei and then other structures that I will not talk about in this lecture. That will become complicated for you to understand. I think it's already quite complicated, isn't it? So we have discussed about the basal ganglia and amygdaloid nucleus. We have discussed about the hippocampus. And now finally let's talk about the cerebral cortex, shouldn't we? So yeah, let's put the cerebral cortex in the picture. So again, the primordial cerebral cortex. Now what will happen that this ventral pole, it will grow ventrally. This ventral pole will grow ventrally to form frontal lobe. This dorsal pole, it will actually form the temporal lobe. It sounds counterintuitive, but it's very logical that it is actually going to form the temporal lobe. How? Because this will fold downward like this, and temporal lobe will come to lie here. It's here at its actual position, and then here a lobe will grow outward to form the occipital lobe, and parietal lobe is somewhat here. This original part actually will form the parietal lobe. So again, it's very easy to understand. Let's just watch the animation so it will become clear. So you see what is going on here. So here if you focus again, that here this posterior pole is going to form temporal lobe, this pole is going to form frontal pole. So temporal lobe was formed here and it rotated like this and comes to lie here at this position, and occipital lobe will form backward. like this. So here I have just removed the one cerebral cortex. Again, it is bilateral structure, or should I put it back? Let's put it back so you really understand it. So here I have put both of the cerebral cortices. I think you can understand that. And please note that the hippocampus, dentate gyrus, the dentate gyrus, and the initial part of fornices, they are actually inside the temporal cortex. They are hidden inside the temporal cortex. If I can show you properly, you can, I hope you can understand. That's why I made them slightly translucent so you can see these structures here. I hope you can see that. And neuroanatomy is interesting. I don't know why people find it confusing. It's very interesting. Now what actually happens that we have discussed already about the formation of fornices and hippocampal commissure. Along with that, some fibers, they are connecting the right and left. They are connecting the right and left cerebral cortices as well, like this. And this bundle of fiber which is connecting most of the right and left cerebral cortices, this is actually called corpus callosum. This is called corpus callosum. So here you can see this is called corpus callosum. So corpus callosum is actually another type of commissure. It is the commissure of cerebral cortex. So we have discussed about the commissure of hippocampus, the hippocampal commissure. We have discussed about the corpus callosum. And finally, there is another commissure which is actually called anterior commissure, which connects the temporal lobe. So these two temporal lobes are connected. Let's hide one of the cerebral cortex. So if I hide one of the cerebral cortex, you can see that here from the temporal lobe, some fibers are going to the other part, the other side of the temporal cortex. So from the left cerebral cortex, nerve fibers are growing and they are going towards right temporal cortex, and from right temporal cortex fibers, they will grow and they will move toward left temporal cortex. So you see like this, and they are actually passing through the lamina terminalis. This is the lamina terminalis. They are passing through the lamina terminalis. Even some ventral fibers of corpus callosum, they are also passing from the lamina terminalis, but of course this 3D model is not anatomically correct, so you don't see it depicted here. So yeah, I think that's all about the development of cerebral cortex and hippocampus. Yeah, you see the indicium griseum here on the top of corpus callosum. So yeah, that was all about the development. One thing I forgot to show in this 3D model, that's very easy to understand, that from the temporal lobe an extension arises, and that extension, its distal end will grow like this bilaterally, and it will form the olfactory bulb. So olfactory neurons, the first order olfactory neuron there, central fibers, they will grow toward the olfactory bulb, and they will take the sense of smell towards the brain, right? So telencephalon, from the telencephalon, in the end, what we need to know, that from the telencephalon what structures develop. That is most important part of this lecture, telencephalon. This is crux of this lecture, cortex and subcortical structures. So cortex is the modern cortex, which is also called isocortex, and the older type of cortex, which is actually called allocortex. And allo cortex is actually divided further into two parts, but let's just ignore that. So isocortex include frontal lobe. This one is the frontal lobe. Um, Parietal lobe is on the other side. You see parietal lobe, temporal lobe, and what else is left? Yeah, our beloved occipital lobe. And from allocortex, what is that? Hippocampus, which include hippocampus proper, dentate gyrus, and indusium griseum, right? And then we have olfactory bulb and olfactory tract. Olfactory bulb, this distal end, I have told you that distal end is thickened. It is the olfactory bulb, and its proximal end, which is attached to the brain, that is olfactory tract. So olfactory bulb plus olfactory tract. You see, only two cranial nerves, they develop directly from brain, so much so that these are not truly considered as cranial nerves. These are considered as extensions of the brain. So these two cranial nerves are first and second cranial nerves. So first cranial nerve is olfactory nerve, which is developed from telencephalon. The second cranial nerve is optic nerve, which developed from the diencephalon, and both of these are the part of prosencephalon. So first two cranial nerves, these are extension of prosencephalon. They develop from the prosencephalon. So we have discussed about these. Regarding the subcortical structures, we know that basal ganglia develop. Basal ganglia, the anatomically classified basal ganglia, because subthalamus and the substantia nigra, they are not anatomically the part of basal ganglia. These are physiologically or functionally the part of basal ganglia, because they have lots of connection with this basal ganglia proper, or anatomical basal ganglia, that they are physiologically or functionally considered as a part of basal ganglia. So it includes the caudate, putamen, and globus pallidus, and along with that amygdaloid nucleus also developed from it. So that was the crux of this lecture. Finally, let me tell you that initially the brain is quite smooth. You see, it's quite smooth. But what happens that as the brain grows further in the limited space of the skull, what happens that it is thrown into folds called sulci and gyri, like this. It is thrown into folds called sulci and gyri, and it increases the surface area of the brain. So what is the sulci and gyri? Actually, if you see that this is the elevation. So if there is elevation of gray matter, this is gyrus. This is gyrus. Then if there is a depression, this is sulci, sulcus. Sulci is plural and sulcus is singular term. So again there is gyrus and there is sulcus. So this is a collection of sulci and gyri. You see, if you see the brain, you will see that it is not smooth. An adult brain, of course, a fully developed brain, even the child brain, I mean the fully developed brain, you will see that it is thrown into such folds that there are elevations and there are depression. Elevation are gyri, depression are sulci, and of course there are fissures as well. There are certain fissures which demarcate different lobes. There is the central sulcus and precentral, postcentral sulcus and postcentral gyrus. All these details you will learn in neuroanatomy. But from this lecture, I think you have learned something. I hope you understood. If you are still confused about anything, please ask in the comments section below. So that was all about today's lecture. Thank you so much for watching this video. Have a great day. Goodbye.